So I have this, they told me I should give this talk. I'm not sure if this is the best idea. I will start to talk about the general feelings about Paclitaxel and Sirolimus. Most people say that Paclitaxel has a smaller therapeutic window than Sirolimus has. If we compare the therapeutic dose for systemic treatment for the respective indications, so we have roughly 300 milligrams for a single dose of cancer therapy when using Paclitaxel, and we have randomized data from Ron Wexman's group about oral Sirolimus for restenosis prevention. And the tolerated dose and effective dose for Sirolimus was 2 milligrams per day oral use. So if you compare it with the amount of drug we have on a balloon, so in a typical 3.0 by 20 DCB has roughly half a milligram of Paclitaxel, and for Sirolimus we have different concentration, but it's in the same area. If you look at the peripheral balloons, we go higher in the single milligram area. And if you compare 2 milligram oral with Sirolimus with, let's say, 1 or 2 milligram on a balloon, then you have the systemic dose already applied with a Paclitaxel, with a Sirolimus DCB in peripheral arteries. With DCB, you are far away from the systemic treatment. The next thing we have to talk about is cytotoxicity. It is true that Paclitaxel have dose-dependent, concentration-dependent increase in cytotoxicity, as shown in this famous paper by Wesley and colleagues, whereas Sirolimus, with Sirolimus, the level of cytotoxicity is stable over the local concentration. However, you have to be aware that with the application of Paclitaxel by DCB, we have very homogeneous drug distribution, not as high peaks we see in the near the stent struts. So in reality, we reach with Paclitaxel also tissue levels clearly below 100 nanogram per milligram. This means in this lower range, 10 nanogram per milligram tissue and lower, we have a similar cytotoxicity as with Sirolimus. So therefore, I do not agree with the statement the one is cytotoxic, the other one is cytostatic. For DCB use, at least, this is not true. The next important thing we have to be aware, if we discussed about both drugs for DCB use, the beauty of Paclitaxel is that it has an irreversible binding to the microtubes. This means if you have the cell has contact with Paclitaxel, it doesn't divide anymore. With Sirolimus, we have a reversible binding to the mTOR receptor complex, and this means an efficacious DCB requires some kind of imitation of the controlled drug release of a drug-eluting stent. So and that's the reason why creating a Limus-coated balloon is not that easy as creating a Paclitaxel-coated balloon. Another word of caution, it's not only the coating composition, it's only the way how a balloon is coated. This is our tissue concentrations over 28 days for the same coating composition, our original coating with Paclitaxel and iopromide. You can create crystalline coatings, and this is the curve on the left side. And you can see that we have a very high drug persistence over a long period of time. You can use the same composition and change some of the parameters, and then you have an amorphous coating. And with this amorphous coating, you have a deep decline of the tissue concentration. So this means it's not only the excipient, it's also the way you coat a balloon. And on the right, we have another version of the Paclitaxel iopromide coating. This is this white DCB. We have seen this yesterday. And they also created a crystalline coating, but with very small crystals. And small crystals have a better solubility, and that's the reason why here we have also, despite the fact that it's crystalline, we have a fast decline of the tissue concentration. So the discussion about paclitaxel or iopramide is only one piece in this discussion. The coating composition and the way you coat a balloon are another parameters that play a role if we discuss efficacy of DCB. This is a relatively old slide where we summarize the available preclinical data on different sirolimus-coated balloons. And as you can see, the initial drug intake is very good with all those types of DCB. But at 28 days, there are very big differences in the tissue concentration. So this need for a long-lasting drug effect is not seen with all the DCB. And this is the published tissue concentration of the Magic Touch Sirolimus DCB. And as you can see, already at 14 days, most of the drug has gone. And this may, or explains, in my opinion, why in the direct comparison with a Paclitaxel DCB with a large database, the sequence, the non-inferiority could not be reached. Late loom loss was 0.32 with the Magic Touch and 0 with the Paclitaxel coated balloon. And this also has been shown that this reduction in late lumen loss by increasing dissection volume has only been seen in this trial with the Paclitaxel coated balloon, but not with the Sirolimus coated balloon. But this does not mean that Sirolimus does not work. It's in this situation, we have Sirolimus balloon with very fast decline of the drug in the tissue. This is a publication from our group 10 years ago where we had a large basic research program and Sirolimus coating compositions from amorphous coatings till highly crystalline coatings. And also here we could see that with very similar coating compositions, the way the Sirolimus was at the end of on the balloon surface makes a big difference in terms of acute tissue transfer and persistence of the drug in the arterial wall. This is a very nice paper from Victor, where he could show that with this high crystalline composition, which I showed you in this experimental data, you can see the crystals and can also see the persistence of the drug in the vessel wall. And we did some smaller mechanistic randomized trials. This was together with our friends from Malaysia, where we compared the Paclitaxel-coated balloon with these highly crystalline Sirolimus-coated balloons. And in the instant restenosis at 6 months, angiographically, there was no difference between the two drugs. This has also been confirmed in a larger trial from China, also comparing the same Paclitaxel and Sirolimus-coated balloon. But we also investigated both types of balloons in de novo disease, and this is the publication from the Malaysian data. And here we found that both were efficacious. Late lumen loss was 0 with the Paclitaxel-coated balloons and 0.1 with the Sirolimus-coated balloons. The only significant difference we found was that late lumen enlargement at 6 months was significantly more frequent with the Paclitaxel-coated balloon than with the Sirolimus-coated balloon. So this means we have to also differentiate between instant restenosis and the treatment of the novo disease. And we did a parallel trial in Germany and Switzerland and found very similar outcomes. This effect of late lumen enlargement with Paclitaxel-coated balloons is due to the fact that we have an increase in total vessel area and potentially some influence on the plaque volume. And one of the reasons for this increase in total vessel area is that Paclitaxel accumulates in adventitia. And this is the reason why we have this Klargov-like effect of increase in total vessel area. Sirolimus is more similar distributed between all parts of the wall, which is also not a bad thing, I would say, but it may explain why we see with Paclitaxel this difference in late lumen enlargement. Now we have solution. I think we are all very grateful that both trials has been positive, because otherwise we would now in a very bad mood here and would not think about making this meeting to a permanent meeting. But I think we need more data, more information on it, and at the end of the day, there will be the question, has it the same efficacy as Paclitaxel-coated balloons in the treatment of de novo disease or not? This is an open question, and I think we have to wait for further trials in the future. So my short summary is that the reversible binding of Sirolimus versus the irreversible binding of Paclitaxel makes Paclitaxel to an easier-to-use agent for drug-coated balloons. But we have to be aware that it's not only the question which drug, it's only the coating composition, the method of coating, which has a major impact on the question if a DCB is efficacious or not. Potentially there are differential indications for Sirolimus or Paclitaxel, but it's, in my opinion, also a totally open question, but we have to investigate this. It looks like that late lumen enlargement is more Paclitaxel specific than for Sirolimus. The question is, will those differences in angiographic outcomes in de novo disease at the end of the day translate in different clinical outcomes? That's also an open question. And of course, that is sure we need longer follow-up and we need trials powered for clinical endpoints for the different types of DCB. Thank you very much.